I'll get going. Yeah, I just wanted to say thanks a lot, Daniel, and thanks everyone for attending. It's really been a lot of fun for me to be a part of this group. I have to say, I really don't feel myself that much of an expert in retinal metabolism or neuronal metabolism. I really wasn't trained in this space, and it's just something my lab sort of drifted into. So thank you for accepting me as part of your group, and at the same time, maybe take it easy on me. I think so. Um, so I'm going to tell you about retinal ganglion cell metabolism today, and hopefully get some feedback from all of you all. But really, the thing that drives my lab is an interest in neurodegeneration. Oh, and I should say, please feel free to interrupt me at any time. I tend to talk very quickly if no one says anything, and even more so over Zoom. So please, either interrupt or raise your hand if I see that, if you have any questions. And so, like I said, my lab's very interested in neurodegeneration, and over the last few years, we've actually been using retinal ganglion cells as a platform to study neurodegeneration. Obviously, there's medical context for this. RGC degeneration represents the most common reason for people to be irreversibly blind. This includes a family of diseases like glaucoma, like trauma, stroke, inflammatory diseases, and multiple different genetic disorders. And currently, there's no treatments that really target the retinal ganglion cells themselves. And so that's really the major goals of our lab are to find new avenues for neuroprotection and regeneration in these different contexts. And again, using ganglion cells as both a relevant medical target and a screening platform to perhaps find panacceas that maybe would treat in other diseases as well. And so just a few important things to realize about retinal ganglion cells. I realized that within this group, photoreceptors are a major target, RPE is a major target. Ganglion cells are a problem because they're a very minor proportion of the retina, but they're a very diverse group of cells. So in the mouse, for example, we have about 45 different types. And they all have different susceptibilities to degenerative stimuli. So in the field, and what I'm going to be talking about today, optic nerve crush, which is an injury to the optic nerve just behind the globe of the eye. It damages the axons and it leads to death of 80% of retinal ganglion cells. So it's been appreciated for more than a decade now that those different types of ganglion cells have different survival potential. And this is just some work from Jin Duan's postdoc in Josh Saines's lab, where they performed optic nerve crush in a few different transgenic lines. And what they noticed was that the alpha RGCs and the IP RGCs, intrinsically photosensitive RGCs, are very well surviving. And a few years down the road, Nick Tran and colleagues, again, in Josh Saines's lab, really took this to the fullest extent, where they looked at all the retinal ganglion cell clusters by single-cell RNA sequencing. And they performed sequencing over the days after crush. And what they identify is really that each ganglion cell type, it basically exists on this continuum of resilience or susceptibility to degeneration. So if we're all the way to the left of this graph, this first class, which is the on-sustained alpha RGC, survives about 95% after optic nerve crush. And if we go all the way to the end of this graph, I believe this is the F mini off RGC, these only about 2% of them will survive. And so this has become another caveat to the platform of screening persistence and death in retinal ganglion cell types. A big question in the field has become what makes these outcomes different? Is there anything we can learn by studying intrinsic differences between these cell types? And so that's something that we've been looking at as a lab. And I'll talk a bit about that as far as metabolism is concerned today. But the other thing, yeah.
If you have different noxious stimuli for retinal ganglion cells, are they all sort of targeting the same more vulnerable set of cells?
That's actually a great question. And for the most part, it was thought that the alphas are like this uber surviving type. We've recently started working in an ischemia model, basically a Rose-Bengal photocoagulation, and there the alphas seem to be quite susceptible. So we think that the different types of degenerative stimuli probably do function a little bit differently. The reports of glaucoma are that glaucoma and optic nerve crush operate similarly. So the IPs and the alphas are well surviving, the on-off direction selectives and similar are poorly surviving. But I do think in different contexts, like perhaps Leber's or ischemia, like I mentioned, there will be differences there. So that's a very good point. Um, so yeah, this idea of what's different between these cell types has been very attractive because it's something that you can kind of dissect pretty in a relatively straightforward way. But a question that's really bothered me since this was, talked about is when you have these outcomes that are 50-50, a coin flip, who's deciding which of those ganglion cells live and die? Because ideally, those are all the same family. They have the same transcriptome, the same other ohm. So what decides whether or not those types die? So these two questions have really driven our research, and we thought that metabolism could be one of these deciding factors because that can be a little bit different even within the same genetically similar cell type. And another reason that we had for this is if you look at the axon regeneration field, so this is another reason people started to gravitate towards retinal ganglion cells, is because their axons make up the only pure white matter tract in the CNS. So the optic nerve is a unidirectional, only white matter portion of the CNS. And so that's depicted here. So in this image, the retina would be over here on the left side, and the brain would be all the way over, maybe a few meters to the right of your screen. And in this case, the optic nerve crush occurred at the asterisk. And this paper from Kevin Park, when he was in Zhigong He's lab, identified P10 knockout as one of the first intrinsic factors that can regenerate retinal ganglion cells. And so, importantly, P10 is regulating mTOR pathway, basically. So knocking out P10 will basically just amplify up the mTOR pathway and promote just basically excessive cell growth is the way that we think about this. And going back to this Duan paper, if you look at different types of ganglion cells within the context of P10 knockout, what we see again is that the alpha RGCs are very regenerative in comparison to other types of RGCs that are still surviving. So this is quite important. And in our mind, the thought was that maybe metabolic potential is what's regulating this difference in the ability to respond to a growth stimulus. So we're thinking that the alpha RGCs perhaps have a surplus of biomolecules that allows them to respond to a growth signal, whereas the other ones that survive the degenerative stimulus are kind of just persisting but not having the ability to grow. So that these are some of the reasons that we started to study metabolism. And so before I get into the actual experiments, I just want to describe what we're doing. So I've been really excited about some of the work that's been shown in this group because you can do metabolomics and you're putting these probes in to measure pH and oxygen levels and all these other things. It's really difficult to put these types of experiments towards retinal ganglion cells because there's so few of them and because there's so many different types. It's really hard to really assay what's going on. So what we've set up is in vivo imaging of retinal ganglion cells through a relatively straightforward approach. Basically, we have a 2-photon microscope, and we set up basically a cover glass that corrects for the cornea a bit, so that we can image through the pupil, basically. So this is just a very fancy way of doing a fundus image. And this is an example of the type of the quality that we can get. It's not quite what you see with in vivo imaging through the skull, because you do have aberration going through the lens, but we can resolve individual ganglion cells. We can see fascicles of axons. If we localize to the nucleus, we can track individual cells quite nicely. And so how do we turn this into something that's actually useful signal? So what we're doing is basically taking an ATP biosensor. So this is called A-Team. And then we can measure out ATP levels directly in vivo. And so the way that this biosensor worked is it's a FRET-based biosensor. So it's a CFP and a YFP pair with a subunit from bacteria that binds to ATP. When you have ATP bound, the CFP and the YFP come into close association with each other, and you get the so-called FRET effect. So your fluorescent signal will be more yellow. If there's no ATP bound to the biosensor, you have more of a cyan color. We make this more confusing by pseudo-coloring the cyan in green and pseudo-coloring the YFP in magenta. But essentially, if I show you something with with a magenta signal, that's going to be higher ATP. Something with lower signal is going to be green, and that'll be lower ATP levels. And before I get too deep into this, I should acknowledge that Zilong Wang is the MSTP student that drove most of this project with a lot of really great technical assistance from a technician in the lab, Chris Zhao. And so to validate the sensor, really, this was one of the earliest experiments Zilong did. His mouse died while he was imaging it, and instead of getting upset, he turned lemons into validation of the biosensor. So if you think about it, a dead mouse is a very good way to change the ATP levels in ganglion cells. So he kept imaging this mouse after it overdosed. And about 10 minutes in, you could start to see ATP levels change. And by about a half hour, ATP levels are pretty much crashed. And so this was really good for us to know that the ATP levels are actually being measured here. We think we're actually looking at a true signal within this sample. And so the first image I showed you was actually tweaked at baseline to show subtle differences in ATP. And that's what we're looking at here. We repeated this experiment in multiple different eyes and multiple different mice. And we get this normal distribution of fret ratios, which we're using as a pseudo representation of ATP. The important things to note here is that we have a normal distribution that is not biased. So that makes us think that our biosensor is likely not up against a ceiling. So we do have the ability to measure higher ATP and lower ATP. And so I think that will be important for some of the experiments I'll show you going forward. And so we then look to see if this is a reproducible trait of the cells. So we image the same cell a couple times over at different days. And we get a reasonable relationship between ATP levels on one day to the next. So this is about a 4 to 7 day follow-up time. So we do think that we're measuring an intrinsic property of the ganglion cell. And then going back to that question I set up earlier about types of ganglion cells, we could take these samples in vivo, dissect out the retinas after fixation and find the same imaging spot back, and then perform immunostaining for different ganglion cell subtypes. And so throughout this talk, I'll be talking about CART, TBR2, and SPP1, or osteopontin. These label the on-off direction selective ganglion cells, the intrinsically photosensitive ganglion cells, and the alpha RGCs. And so what we found with this is that these lower, greener, lower ATP RGCs tend to be enriched for the alpha RGC, which was a little bit surprising to us because the idea was that these are going to be really metabolically active. And so we wanted to see what's really underlying these differential levels of ATP at baseline. And the very superficial way of thinking this is that you could have differences in consumption, or you could have differences in production, or some combination of both. So we started with consumption because that's something that we've worked on previously, basically modulating the activity of the ganglion cells. So the reason that we think activity is the major driver of this is that action potential spiking, maintaining a charge across your plasma membrane is one of the most expensive things for a neuron to maintain. And ganglion cells have different firing properties. So this is some experiments from Thomas Euler's lab where they present a chirp stimulus. And basically, if you just think about the area under these curves, so each of these lines is a ganglion cell type, you can see that the area under the curve is very different for these ganglion cells. These are sustained ganglion cells that are presumably firing a lot in response, where on-off cells tend to be a lot less firing to this response. So we would presume that these cells here are consuming more ATP. At the same time, Sean McCracken in the lab has previously worked with a calcium biosensor. This is set up the same way, so high calcium's magenta, low calcium's green. And we see this differential distribution of calcium levels under homeostasis. So this is with a lot of caveats due to our imaging conditions. But we know that this is also a population enriched for alpha RGCs, these high calcium cells. And so what Zilong did was he injected activity modulators into the retina and recorded the calcium response and compared that to the ATP response of manipulating activity. And so what I'm doing here is showing that same calcium sensor. We've pseudo-colored it differently here to make it discernible from the A team. But essentially, cyan is low calcium, red is high calcium. And what we're doing is injecting strychnine and bicuculin into the retina, which will disinhibit the cells by blocking glycinergic and GABAergic receptors on ganglion cells so that they will spike more. And what you can see here is that calcium levels do indeed go up quite high. And what you're going to be looking at here, these line traces are individual ganglion cells, and they're pseudo-colored where cold colors are a low level and hot colors are a high level, all anchored to the pre-time point. And what you notice is a lot of mixing here, where we have some low cells that become high after the change in disinhibition, and that's all graphed out with single RGCs here. However, when we do this with ATP, we see more of a concordant response where the whole population is shifting together. I apologize, this image is a little bit blurry, but basically after we inject strychnine bicuclian, what we do expect to see is happening where ATP levels are dropping because activity is going up. But what happens is we don't really see much of a change in rank. So low ATP cells remain low and high ATP cells remain high relative to others. Although in spiking, there's a lot of mixing here. So it doesn't look like this would explain a cell suddenly becoming high spiking and then suddenly becoming low ATP. It doesn't really mesh with that idea. So I'll show some evidence for this later, but what we think is happening is you actually have a local overconsumption of things like oxygen and perhaps glucose that is driving down the overall ability to make ATP for the milieu, but not so much changing individual cells that are themselves spiking a lot. And we do the opposite experiment where we're blocking glutamate receptors with MBQX and AP5. This is going to block activity, and we show that with calcium levels here. All the calcium levels tend to go down. You do have these transient spikes in calcium levels that we presume are off RGCs, but they go away after a baseline is reached at about 10 to 15 minutes. And you see a concomitant similar change in ATP levels. They're not as strong, but ATP levels slowly will rise in this condition where we're silencing the circuits of the retina. And again, we think that this is overabundance of oxygen and perhaps glucose that's driving this, although it could be the fact that all cells are now just consuming less ATP because they're not active anymore. But you see that same thing where the low cells remain low and the high cells remain high. And so we don't really think ATP consumption is driving these differences. Yes.
Yeah. So have you tried quantifying that at the level of individual? I guess you can't do it for the same cell. Like the delta for a particular class, the delta of the signal for calcium versus the delta of the signal for ATP, and really plotting that out next to each other.
Yeah, we'd love to do that, and there are actually ways to do that. So we could perform 3-color imaging, where we use like an R gecko to measure the calcium alongside the ATP. I think that would be the easiest way to do it, because even with the subtypes, like for instance with the alphas, you have on alphas and off alphas, and we think with these treatments you get kind of differences in their response there.
Yeah.
So we think doing it within the same cell is the way to go. Fortunately, I think you didn't review our paper because they didn't ask for that. So,
No,
so we haven't
no.
done that experiment yet. Although I am very curious about it, we just haven't set that experiment up. Yeah,
Okay.
yeah. So yeah, then we went into production strategies, and honestly, we've basically injected everything you can think of that would mess with metabolism in a cell. I'm just going to show you a curated list of effects related to oxidative phosphorylation. I will say we injected 2DG and didn't really see a response, so we kind of ruled out glycolysis. The thing is that, you know, there's a lot of problems there. If you did affect glycolysis, you would affect oxphos anyway. But we're going to jump straight into the mitochondria. So what we did was to check if mitochondrial respiration obviously is going to be important for this, but we wanted to see how important it is across different RGC types. We started with complex 1 by injecting rotenone. We also injected other inhibitors with this that I'm not going to show. And what we saw was actually something quite curious. So I'm going to play a movie here. Each frame is about a second long, and this movie is represented in this trace here over to the right, where again, each line is a ganglion cell and time is on the y-axis here. And what you'll notice is that a subset of ganglion cells are affected by this. So you see a subset of ganglion cells that are turning green. I wouldn't say they're in a mosaic, but they're definitely spaced out. And this looks like certain types of ganglion cells are specifically inhibited by rotenone injection. And so we went on and did our immunostaining with this. And here what we find is that the alpha RGCs, again, are more inhibited by rotenone. And we added a fourth type here to the graph, the TBR2 SPP1 double positives. These cells are the on-sustained alpha RGC. Yeah, sorry, just a second. Someone's knocking at my door. I want to make sure it's not an emergency. Yeah. Hi. Yeah.
I'm seeing in your Facebook, there's a perfusion this morning. I think they got scared off and didn't clean up.
Oh, I'm
So-
sorry.
But just tell them that they
Yeah.
they left a diaper with blood
Okay.
there on it. Anybody?
Yeah.
And see that they can.
I'm actually giving a seminar right now, but yes. But yeah, I'll let them know. Yeah. Apologies, I had a surprise IACUC inspection, so it sounds like everything went okay, but I apologize for that. Yeah, so basically the unsustained alphas are the most affected by this, where the alpha RGCs are also specifically inhibited. The next question we had was this: is this specific to complex 1, or is it because we had a partial inhibition of the electron transport chain? And so we injected TTFA to block complex 2 specifically. So this would be FADH2 dependent instead of NADH dependent. And we get a similar phenotype here. It's a little bit more broad. So you can see that the whole population is dipping, but you get a stronger dip in the alpha RGCs again. And again, the on-sustained alpha is the strongest affected. Then we checked the later steps of OXPHOS, where we're looking at complex 4 and also ATP synthase. We used antimycin A, and we're going to show data for using potassium cyanide. And here again, we get this trend where all ganglion cells start to be recruited to losing ATP, but you get a stronger effect in the alpha RGCs.
Can I ask a quick question?
Yeah, sure.
This could be kind of naive, but can you remind me what is special specifically about the alpha ganglion cells? Like what so far we know about their function and how they differ from the rest of the ganglion cells?
Yeah, sure. So the alpha RGCs are actually quite conserved. There's many species have an alpha retinal ganglion cell. And the thing that's kind of important about them is that they're generally found in high acuity visual areas. And the analog to the midget and parasol in a human is actually the alpha in the mouse. So that's what makes them sort of interesting, is that they're actually used in almost pixel perfect vision in your fovea. So they're coupled to single photoreceptors there, not so much in the mouse, but you do have a binocular zone in the mouse that's used for things like prey capture, and the alphas are enriched in that zone.
Okay, so they're specifically located near the central retina then?
They're not specifically there, but they're very enriched in those regions. In the mouse, at least, most ganglion cells exist everywhere, but they are higher density in the area that would see the central visual field.
Awesome. Thank you very much.
Yeah, yeah, sure. Okay, and then I still don't feel like we figured out this question that we set out to answer. But we did try going back to the first experiment that I showed you, where we accidentally killed a mouse. And if you look at this 10-minute image now, you might pick out these cells that are turning green faster than the other ones. So we revisited this concept, but on purpose this time. And so for these experiments, we overdosed the mouse. We set up a pulsometer to see what the heart is doing at the time, so we can tell if the mouse is dead yet or not. But this is the image that we got, and we think it's pretty cool because you can see cells slowly recruited to this low ATP state. It's not just a cliff that all the ganglion cells fall off. And the thing that was most interesting about this is when we graph out all of the different ganglion cells, we basically see different phases of ATP loss, as I indicated. So bradycardia sets in here. This is when the heart's not going to be pumping as efficiently. And then the heart stops at asystole. So we think that this early phase of ATP loss is probably representing a loss of oxygen in the system. And then that is represented in this plateau here, which is interesting. So these ganglion cells at the bottom are basically OXPHOS dependent to main their ATP. These ganglion cells, oh, I forgot to ask, people can see my pointer? Yes?
I think it might be better as a laser. I can see it
Oh.
if you move it.
Okay, my bad. Yeah, I always have trouble setting this up and then getting back into
Oh yeah, I mean,
Zoom.
people can ask questions if they're not sure what
Okay,
you're talking about.
sure. And so we think in this plateau region, the cells at the bottom are obligate OXPHOS. The cells in the middle are probably hybrid, so they can rely on glycolysis and OXPHOS to make their ATP. And these cells at the top are probably able to maintain ATP, relying only on glycolysis.
Wait,
Nancy, go ahead.
oh, thank you. Philip, are you perfusing the solution the retinas are bathing in with oxygen?
This is a live mouse or a dead
Oh,
mouse.
this is
This is in a mouse.
Oh,
Yeah.
dead, but in the mouse. Sorry,
Yes.
I thought it was ex vivo.
Yeah, no, we start in vivo and then we make it ex vivo, but we don't take anything out. Yeah.
Okay.
Yeah, it's a weird prep. I think it makes sense, but it's good I'm hoping to get some feedback. But
No, I just wondered if
Yeah.
you could increase oxygen tension and see if that was due to loss of oxygen.
Yeah,
Yeah.
we're trying to get into a system where we can actually control and titer the oxygen levels to the mouse. So that's something that we're setting up to do, but we haven't got there yet. Yeah.
So this response pattern looks, to me at least, pretty different than some of your inhibition experiments with, you know, antomycin or KCN, which I guess themselves look different from each other.
Yeah.
But those would essentially, something like KCN simulates the loss of oxygen because it blocks the, enzyme that consumes oxygen. So why then don't you have this sorting of these subpopulations in the same way as you do in your recently ex vivo mouse?
That's actually a very good point. So I think the antomycin A difference is that it's an irreversible inhibitor. So even it gets washed out of the system, it's still bound and doing its thing. So that's why we have a floor that never gets relieved. For the KCN, that's actually a very good question that we hadn't thought about. Ideally, this shouldn't have made a problem, right? Like those cells that don't need oxygen should have been able to persist. Yeah, I don't really have a good answer for that, to be honest.
There are a
It-
lot of things that happen after death. That is for sure
yeah,
different than directly treating with KCN. And given Rob Linsenmeyer's pretty recent talk about lactate and acidity, I do wonder if a lactate buildup, well, it's going to happen both with KCN and after death. Okay, I'm not sure. There's a lot going on here. So but
I would also I don't know how clean cyanide is because I thought it does more than just hit complex 4. But if anything, that may be doing causing other problems.
The cyanide is easier to trust than the antomycin just because your first two inhibitors have the same bounce back effect.
Yeah. No, that's a very good point. And the time range we're looking at, I don't think this is going to be a rundown of metabolites. Like that should not be operating within 5 minutes. So this should indicate something about oxygen. It might also be that the oxygen levels are slowly depleted and the cells might be able to, compensate with that kind of inhibition where KCN is just going to remove it immediately. That's another way I could think of explaining it.
Or interactions between different retinal cells are unable
Yeah.
to occur in the presence of KCN, but could occur to some extent in the recently dead mouse.
That's a good point. Yeah, all this is ignoring the glial cells and the vasculature. All these changes could be compensating in some way. Yeah, we would like to get to the glial side of all this at some point. And yeah, so basically what we think is happening is we're sorting out the cells that are OXPHOS obligate, hybrid, and glycolytic. And then we think that when we lose this plateau, that's actually the local metabolites start to run down. So this is about, I didn't indicate the time here, but this is about 30, 40 minutes after death. So we tried to address that question. We happen to have a glucose biosensor. And so this is what the glucose biosensor looks like in ganglion cells. We were already quite curious about this too, because we see different resting glucose states in ganglion cells, which is intriguing to us. We haven't been able to explain this, and this is one of two stimuli that actually changes glucose levels. So if we look at 3 minutes when ATP is starting to drop, we don't really see a significant change in glucose. But if we look at 30 minutes on, glucose levels within the ganglion cells are starting to drop. So we think that that is what's causing this tail at the end of the post-mortem loss of ATP. And so essentially the way that we're thinking about this is we have these different types of ganglion cells that are really existing on a continuum of oxphos dependency to glycolytic. I wouldn't say dependency, but they have the ability to maintain without oxygen. And then these cells out here, I don't know what's going on. There's likely very little glucose left in the system and very little oxygen, but they still seem to have ATP. I would say there is like a constant slight drift here that is going down, but they're still rather high for how long post-death they are. So I would say, again, we haven't really answered what's setting this differential thermostat, but we do understand a little bit more about the systems that ganglion cells rely on to make ATP. So we wanted, again, to look at that question of what sets this different ATP level at baseline. And one of the things that we thought might explain it is mitochondrial calcium levels. Because I've shown you that work that Sean did looking at cytoplasmic calcium. We thought that stimulating the mitochondrial calcium could be one way to stimulate the TCA cycle and stimulate ATP production. And so this is work that Sean has published, and we've also recently submitted, where previously we looked at cytoplasmic calcium. We've localized that calcium sensor to mitochondria. And what we notice is that the mitochondria calcium levels tend to track relatively well with cytoplasmic, which is what you would expect. It's thought to be somewhat passively downstream of cytoplasmic calcium.
This is in the matrix?
Yes, this is a coxate fusion protein to localize the mitochondria. And then when we do our typing analysis on this, indeed, what Sean finds is that the alpha RGCs and the IP RGCs actually are somewhat higher than the non-alpha non-IP RGC population. I would say one caveat here is if we pull out the on-sustained alphas, these significant levels go down quite heavily. So it looks like the on-sustained alpha is driving this difference in mitochondrial calcium pretty heavily. But the thing there is that this would indicate that our low ATP cells actually have higher homeostatic mitochondrial calcium levels, which goes a bit opposite the hypothesis we were thinking. The other thing we looked at was mitochondrial content. So Shelley Zhu, an undergrad in the lab, did an immunostaining panel of retinal ganglion cells and a number of different mitochondrial proteins, staining for all ganglion cells with RBPMS and the alphas with SPP1. And what she found overall is that the mitochondrial content by immunostaining at least looks higher in the soma of alpha RGCs, which again goes against the hypothesis that they have less ability to make ATP through their mitochondria. That's what makes them more susceptible, and that's what makes their levels lower. So we think they actually have more abundancy of mitochondria. So then what we're thinking now is to look at the mitochondrial characteristics that maybe make them less efficient at making ATP. So we've done some preliminary immunostaining for UCP1 and UCP2. I have to say these images aren't fantastic, but we do see through semi-quantitative immunostaining slightly higher levels of UCP2 in alpha RGCs. So we think that the uncoupling system might explain some of the differences that we're looking at.
I'm sorry, I
yeah.
have one question. So these are a cross-section?
These are in cross-section. Yeah, sorry, I didn't point that out. Yeah, the previous were in whole mount. So yeah, yes.
Sorry, uncoupling this is one of those unfortunate, more of a comment than a question. Having worked on uncoupling proteins a little bit, there's a lot of argument as to whether they actually uncouple or are just proton gradient driven transporters. And so you should keep that in mind. I think there's at least one paper suggesting that they're aspartate transporters, and the science in that paper seemed pretty good. And other papers suggesting that they're fatty acid transporters.
Okay, cool. Thanks a lot. Yeah, we're actually doing some work right now. We just injected the mice last week to overexpress or knock down UCPs, and then we can read out the ATP at the same time. So yeah, perhaps they're not working through uncoupling, but at the very least, we can see if they're maybe underlying some of these differences. Yes. So that's, as I mentioned, we're Mingley in the lab is trying to manipulate UCP. And the other thing, Sean in the lab is setting up some spatial sequencing that we can combine with our biosensor imaging. So that's basically where this is at. We haven't really figured out what the magical thermostat is, but we do see differences in the way that ATP is produced across our GCs. And so getting back to how I started this whole talk, what about neurodegeneration? And so there's a lot of evidence that's tying retinal ganglion cell neurodegeneration to metabolism. So you have a lot of work from Pete Williams's lab in glaucoma and other models that's used NAM and pyruvate and other molecules like vitamin B to supplement and maintain RGCs. Also, Denise Inman's lab has done some MCT2 overexpression in glaucoma and shown that to be protective. At the same time, metabolic signaling seems to be at play as well. So Adriana DePaulo's lab in glaucoma has looked at AMP kinase and insulin signaling. And as I mentioned early on, mTOR pathway is very protective of certain types of ganglion cells and optic nerve crush. So we basically just wanted to say what's happening at the ground level of metabolism, because the theory is that most neurodegeneration occurs through metabolic catastrophe. So that's what we were hoping to see. So we inject our 18 biosensor. We do a pre-image, perform optic nerve crush, and then we're imaging every 2 days for 2 weeks after the crush. And so if we don't have a biosensor in there, if we're just looking at ganglion cells, this is what we can track in vivo. So this is with H2B GFP. By a week, more than half the cells have died. By past 2 weeks, you almost have to just believe me that this is the same part of the retina. And we can make these schematics and say, okay, when did these cells die? And then by comparing this with our biosensor, we can start to ask questions about what's driving cell death or survival. And so pairing this with A-team imaging, what we found is that the surviving RGCs indicated here with green arrows versus the dying RGCs with the red arrows tend to have lower ATP levels. It's all baseline ATP and homeostatic ATP levels. And so that's great. We know alpha RGCs have lower ATP. We know they survive better, so maybe that's what's explaining this. We then performed immunostaining on these same samples to look at the survivors and say which ones are alphas and which ones aren't. And we pull them out of the analysis. We still see that surviving ganglion cells that are not alphas also have lower homeostatic ATP. So we think that the lower ATP level might be actually setting up RGCs to survive better. The other thing that we could do is image ATP throughout the course of degeneration. And what we were really surprised by was that we actually see a spike in ATP. So what we were expecting was that there would be a metabolic catastrophe that would precede cell death. And this would show up with cells becoming extremely green before they would die. But actually what we saw was that peaking around 4 days across all samples, we have this very prevalent spike in ATP levels across the population. And I didn't believe this at first. I actually had Zealand repeat these experiments 3 times. And then we repeated it 2 more times for the revisions of the paper. So I very much trust this data. There is this spike in ATP after optic nerve crush. And so we asked the logical question coming from the degeneration field, let's mess with the thing that goes wrong after an injury. And we tried dropping ATP levels. So niclosamide is an FDA-approved dewormer that also uncouples the mitochondria. And so we treated mice with niclosamide gavage daily, preceding nerve crush, and for 2 weeks after nerve crush, or every 6 days of every week, basically. And what we find is that this is protective. So if we drop ATP levels, that seems to preserve ganglion cells. Surprisingly, so this is about a 60% survival effect or increase in survival between niclosamide and vehicle. We are still doing experiments to confirm that niclosamide, as we're treating, is bona fide dropping ATP. So we need to clarify that that is what's happening. But this is the data we have right now. And so we're trying to figure out then what's driving this ATP shift. And so our thought is that we think AMP kinase might be the driver. So AMP kinase usually canonically responds to a change in ADP and AMP ratio that stimulates the phosphorylation of AMP kinase, and it basically drives a lot of responses that will increase ATP production, increase metabolite sparing by decreasing biogenesis and increasing metabolite uptake. But it's also been indicated, as I've shown in glaucoma, Adriano DePaulo's lab suspects that AMP kinase is driven by cell stress as well. And so we happen to have biosensors for a bunch of these steps in the pathway. We have an AMP kinase biosensor, and we have biosensors for glucose and reactive oxygen species. So this is our AMP kinase biosensor data. I should preface this with the dynamic range on the sensor is only about 30%, so it's not really that robust. But even with this small dynamic range, I hope you can appreciate that this cell here has an elevation of AMP kinase and then is lost. And the same with this cell here, has a slight elevation of AMP kinase and is lost. And if we split our surviving and dying cells and AMP kinase activation, we see that our dying cells tend to have a stronger AMP kinase activation in these crush experiments. Since this biosensor isn't the best, we also looked into our other biosensors downstream, presumably of AMP kinase. So we looked at glucose as well with our glucose biosensor. I apologize, this is a little bit of a messy figure. But what we notice is that in the early days after optic nerve crush, we actually have this upswing of glucose. This biosensor has a 65% dynamic range, so it's also a little bit on the small side. But we see this uptick of glucose in the population, followed by a little bit of a downtick. And the other thing that's interesting here is if we split our cells into dying and surviving, our homeostatic glucose levels are lower or enriched for lower in their surviving phenotype. So this also matches our ATP. And then lastly, we see mitochondrial reactive oxygen species are very heavily upregulated after optic nerve crush. So this is a different type of biosensor. It's an excitation ratio metric biosensor. But here, what we're seeing is these magenta signals are coming from mitochondria and RGCs that are heavily oxidized. And we can see the quantification here. This actually just accumulates over time, so it could be related to cell stress and other pathways outside of AMP kinase. But that's basically what we're seeing. The other thing, since we have the ability to look at it, that we wanted to see is if mitochondrial calcium might also be upregulated, driving more ATP production after injury. So we should say we've already looked at cytoplasmic calcium dynamics after crush. So this was in Sean's paper from 2023. And what we found there was also a little bit surprising in that higher calcium RGCs tend to be the ones that are surviving. So if we look at surviving and dying populations here, the higher calcium cells survive more, or the surviving cells are enriched for high calcium. If we split the ganglion cells into two populations of high and low, what we see is that the high calcium RGCs are surviving 4 times better than the low calcium RGCs. But interestingly, we don't really see a calcium signature for dying cells. So if we look at the dynamics of calcium in a cell before it dies versus when a cell is not dying, they look relatively similar to one another. So we wanted to do the same thing with mitochondrial calcium to see if that might be driving things. Interestingly, we see a similar phenotype where higher mitochondrial calcium is enriched in the surviving population of cells. However, the dynamics of mitochondrial calcium go the opposite of what we would expect if it's driving ATP production. So we actually see mitochondrial calcium levels diminish after optic nerve crush. So this, we think, indicates that it's likely not driving the ATP spikes. Interestingly, though, if we treat mitochondrial calcium, it acts inversely to what we see of this well-surviving phenotype, where if we mimic this decrease in mitochondrial calcium that we see after the crush, so we do this by injecting RU265, we can actually preserve retinal ganglion cells by lowering mitochondrial calcium levels. So this again goes against the idea that ATP driven in the mitochondria is what's detrimental. Calcium entering the mitochondria is what's driving the ATP. So yeah, just to summarize what I've shown you today, what we see is that homeostatic ATP levels across ganglion cells are diverse and a salient feature of the ganglion cells. They seem to correlate with type, although within types there is some diversity. We didn't really get into that here, but there is still a spread of ATP levels within RGC types. For the most part, oxphos is more important than glycolysis, although saying that is really tricky because you don't have oxphos without glycolysis. But I would say aerobic glycolysis is less critical than the mitochondrial production of ATP, where some RGC types are requiring oxphos much more than others. Well-surviving RGCs tend to have lower homeostatic ATP levels, and they're more dependent on oxphos for producing ATP. ATP levels counterintuitively are elevated by optic nerve crush in this degenerative model. And interestingly, we think that lowering ATP with niclosamide is actually protective. We're trying to get at this by measuring ATP within the niclosamide system, but also using other ways to lower ATP, like overexpressing the UCP is one of the setups that we have going. And then lastly, we think that the ATP overshoot could be causing mitochondrial stress, which is leading to degeneration. And this might be downstream of AMP kinase signaling. And yes, the mitochondrial calcium levels are higher in well-surviving RGCs, but paradoxically, the dynamics of making an RGC survive are inversely related to the well-surviving phenotype. So where are we trying to go with all this? So we'd like to see how metabolism is compartmentalized. And so Minglei Zhao, this postdoc in the lab, has set up transscleral imaging. And here we get these really beautiful images where we have subcellular resolution. So we're trying to take advantage of this to look at, say, dendrites versus axons versus cell bodies to see how different compartments deal with metabolism. At the same time, we're starting to express some of our biosensors within the molar glia to see how the glial cells are participating in all this. Another thing that we really are trying to figure out is this metabolic set point diversity, what sets that thermostat? And we've had a lot of trouble getting at this even with all of our biosensors. And so what Sean McCracken has been setting up in the lab as he's about to leave for his postdoc is something we're calling SenseSeq, where we take our in vivo imaging of the biosensor. And Sean's developed a way to section these on phos into 12-micron sections that we're now sending off for MERFISH, so that we're trying to dive into the transcriptomics that underlies this biosensor diversity. And we're very excited to see where that goes. And then lastly, as you asked at the very beginning of the talk, what about other systems? So we've set up glaucoma models and we've set up retinal ischemia models to see if we see the same relationship between ATP dynamics and survival, whether these treatments can be used across the board or whether they're very specific to traumatic or traumatic adjacent lesions like glaucoma or suspected NF1 and things like that. And then once we have these treatments for nerve crush, can we move them into other systems? And we're also doing behavior to see if these things can maintain vision, not just maintain retinal ganglion cells. So with that, I'll wrap up, and I really have to thank my lab. I mean, they're the ones that did all of this work. The people that were critical for the metabolism side are bolded, so Chris Jiao, Zilun Wang, and Shelley Zhu. And then I was also showing some data from Sean McCracken with the calcium story and Minglei Zhao with AMP kinase. I also have to thank funding and all of our collaborators for the projects I wasn't able to talk about. So Ximing Chen's working with us with photoreceptors. Josh Morgan, we're looking at the LGN response to degeneration. Nick Tran and John Dam were doing some activity in neuroprotection. Zhigang He is working with us on AMP kinase. And then part of the iWire2 group and Rachel Wong, we have a story that's not related at all to any of this with retinal ganglion cell primary cilia. So yeah, with that, I'm happy to take questions. Thanks for paying attention, hopefully.
All right, questions. Thank you.
Hi, Steve Abkauer. I have a question. Can you hear me?
Yeah.
It's one question. So the first question is that I was wondering what kind of the niclosamide you were using. Are you using the form that's used for deworming? Because that's not really bioavailable.
Yeah.
But then there's some that have been used, I think, for cancer, which are like derivatives, which are bioavailable.
Yeah.
The dewormer, you don't really want it to be bioavailable because the worm's in the gut, right?
Yeah, I didn't get into that. But we're using the one that is bioavailable, so it's the salt that we then put in corn oil and deliver via gavage. We're trying to find other ways to deliver that's a little bit easier for the mice because the gavage, actually the reagents you need to put the niclosamide into solution actually are kind of toxic as well. So we're worried about side effects of just the vehicle, honestly. But yeah, that's a very good point. We're not using the deworming one. We're using the
Okay.
salt for cancer.
And then just the other comment, maybe, when you do crush,
Yeah.
does axonal transport shut down? And that could maybe shut down that demand for energy.
That's a very good point. So the axonal transport does shut down because the axon is gone. There is still some transport that's residual, but you see actually a pileup of mitochondria in front of the crush site. So that occurs after injury. But work from Adriana DePaulo has done some in vivo imaging of mitochondrial transport in glaucoma, which is a different model, but also affects the axons. You also see a slowdown of transport in that system. So that's a very good point, that there might be a decrease in demand from the non-retinal portions of the cell. Our thought was that a lot of that is probably taken care of locally by glial cells in the optic nerve. But it is true, if mitochondria are building up in the soma, it's possible that is allowing for more oxphos to take place.
How's that taken care of by glial cells?
I mean, the last I was reading about it was oligodendrocytes feeding lactate to axons, but I'm not 100% sure how well accepted that theory was.
Nancy. You're muted, Nancy.
Great. Oh, sorry. Great talk. Really interesting. I wondered if you could couple what you're doing, if anyone at WashU is using FLIO, since, it's not quite clear well, to my knowledge, it's not quite clear how FLIO reflects metabolism, and whether some of your studies could be coupled with that.
So
using
that's
FLIO.
So FLIO is flickered light electro. Sorry,
Lifetime imaging.
It's fluorescent lifetime imaging.
Oh, okay.
Sorry.
Yeah,
I guess it's split. Sorry, not anyway. Sorry.
Yeah, I'm not sure how well that interfaces with what we're doing. I thought FLIO, correct me if I'm wrong, I thought you need adaptive optics for that, or am I off base there?
I just saw, I looked up, they are using it in mice. I'll look it up and tell you
Okay.
what, there are some recent papers where it's been used in mice, and there are some people using, Bob Surgott in ophthalmology at Jefferson is using it on patients to reflect metabolism and changes in metabolism with aging, but it seems like the coupling the information you're getting, maybe not on the same mice, but the same treatments, and then compare them.
Yeah, we've been trying to get into intrinsic imaging. I think that's sort of similar to look at the intrinsic fluorescence of NAD and FAD molecules.
Right, yes.
Yeah.
It is, that's it.
Yeah, with our imaging, we've tried that. We have too much aberration going through the pupil the way we do. We are hopefully getting an adaptive optics system that will allow for us to resolve these things. So that might be something coming down the road. But yeah, currently we can't really see any endogenous signals with the way that we're doing the imaging.
Okay.
Yeah.
Thank you.
Yeah.
Asda.
Yes.
Well, thank you for your presentation. It was actually very interesting.
Oh, thank you.
I have a few technical questions.
Uh-huh.
I wondered about the age of mice that you used through all your experiment. And generally speaking, do you think you would see a sex-specific difference between male and female when it comes to the ATP?
I would say we try to keep our mice between 6 and 8 weeks old at the start of experiments. So some of the data might be up to 10 weeks old here. But yeah, mostly youngish adults is what we're going for. As far as male-female differences, we didn't notice any, but we haven't looked extremely diligently, I would say. But with that cohort of like 11 retinas I showed in the beginning, we did split that by male and female, and we didn't see an obvious difference between male and females, just resting ATP levels. But honestly, that all those measurements are Z-score analyzed, so that's not going to be comparable across mice, actually. So we'd have to go back and check it, yeah.
Okay.
With the raw levels. I think that the issue is there can be a little bit of drift in our system, so it's a bit tricky Yeah, I don't talk about it much at all, but our PMTs, their sensitivity degrades over time. And so we have to do a whole bunch of stuff to try to correct for that. And I'm worried that something subtle like a male-female difference that requires a lot of compiling over time, we might just have intrinsic issues with it. But yeah, short answer, no, we don't know.
Okay.
Yeah.
Another question would be, I noticed in the small short videos that you played that it's kind of the same area. Is there like, are you focusing to record a specific area, or was it different? Because for my vision, I thought it was same area always in the slides that you showed.
Yeah, so essentially, we're a bit limited that we can only image relatively central retina. That just has to do with the angles that you can achieve going through the pupil. And so the way that we set up our imaging is we use the optic nerve head as a landmark, and then we'll go adjacent to that, maybe like 100 microns away from the optic nerve head in any of the cardinal directions. So we might be dorsal to the nerve head or ventral to the nerve head, and sometimes it's the left eye and sometimes it's the right eye. So, that's going to invert. But generally speaking, we're near the nerve head. The imaging field itself is about 500 microns by 500 microns, just to give you an idea about. So it would be roughly 100 to maybe 700 microns from the optic nerve head in any direction, I would say, is how I would define the space.
Okay, so also for the whole mount figures, you also focused on that area, I think.
Yeah,
then.
the way that we do the whole mount imaging is we'll acquire imaging field that represents the area that we can image in vivo, and then we'll pull out 4 fields of view within that area and count those. That's why traditionally our RGC counts are a little bit higher than what's in the literature, because we don't actually sample periphery when we do counts. We only sample the central retina.
Okay, and the quantification was done manually, or is it
This is all manual, unfortunately. Yeah. So
Okay. Okay,
yeah.
thank you very much.
Yeah.
I have at least a couple questions. I think there were a lot of questions that probably came up during the figure, and then I forgot them as you transition to the next figure. So I apologize.
Sorry. Yeah.
No. It's a lot of really cool information. And so it's a good thing. I think since we spoke about the uncoupling agent as niclosamide, the classic story is that uncoupling kills, right? Like people used to try to take it to lose weight and then they died, or at least they overdosed and then they died. So what level of uncoupling are you achieving with these?
Yeah, so that's what I mentioned. We're trying to do the ground truth data now, where we treat with niclosamide and measure ATP in the eye.
ATP might not be a helpful metric of that,
Okay.
because you have been inhibiting literally mitochondrial complexes that generate ATP,
Yeah.
that are used, the electrons are used to whatever, shuttle protons that generate ATP. Yeah, so like if those aren't working, my expectation is that you're seeing these like drops, and then that cell that was dependent on oxidative phosphorylation becomes dependent on glycolysis.
Yeah.
And so measuring ATP as a mitochondrial output is not ideal. It's integrative. And so maybe you should consider measuring either membrane potential. They have floors for it, although I'm sure it's tricky to use those in vivo.
Yeah.
But then, yeah, I don't know, do they have any sensors?
So we have a mitopyruvate sensor that would probably maybe work, or
A mitochondrial
say
membrane
no?
potential sensor would be ideal.
Yeah, we've played around with like dyes like TMRM, and that's just a no-go in vivo. It
Yeah.
barely gets into the
figured-
retina, and you get a lot of signal everywhere else.
Yeah, okay.
We might be able to do that with transscleral imaging, though. I showed that image of the transsclera, but there you get much higher resolution. We might be able to resolve RGCs with that.
Okay. Yeah, it is tough. I do wonder if though there's a fluorescent dye that you can use that will serve you a little bit better for actually measuring membrane potential.
Would acidification of the compartment maybe be a better way to look at the actual protein concentration?
I think if you have a mitochondrial pH sensor, it will serve you well. That could work.
Yeah, so we do have some pH sensors. I don't know if they're calibrated for that level of acidity is the issue.
Yeah.
because most of them are more physiologic and not for hyperacidified spaces. But I think some of them do exist for measuring things like lysosomal pH. So
Yeah.
I can think of something like that.
Yeah. And people, yeah, I think it would be an interesting question to answer. Some of the times where you're seeing, say, more resilience corresponding to less baseline ATP levels.
Mhm.
I'm sorry if some of your data disproves it, and I'm just kind of not integrating it into a whole story in my head. But to me, low ATP levels, and I think you know this based on your presentation, could mean high consumption rate of ATP. It's a cell that is just, using that ATP well to do whatever it does. So is it paradoxical that you might have high calcium and low ATP? I don't think so, because high calcium can elevate TCA cycle activity and can really help generate ATP in a cell that rapidly needs to use it. I think, there are all sorts of configurations, I guess, of concentration of ATP and calcium that I think are okay and not so paradoxical.
Yeah, no, I agree. I think I think we're just thinking of it a very basic view. Like this is high, that should be high. This is low, that should be low. I think the main evidence we have against the activity driving this baseline is when we change activity, it doesn't get rid of the discrepancy in ATP levels, right? So if we silence the circuits in the interplexiform layer, the ATP doesn't, come to one unified level. You still have that gradation of set point. There are obviously other tasks that consume ATP in a neuron. It's just thought that activity is the main driver there, but it
Yeah.
could be like cellular complexity or something like that might be part of it as well.
And how are you silencing activity again? I forget the mechanism there.
We were injecting glutamate receptor blockers and we measure out the calcium level, and the calcium plummets to a uniform low level, non-spiking is what we're assuming there.
And that was affecting in-cell ATP, like within the same cell, but not between different cell set points. Is that right?
Yeah, let me go back to that. So basically what we see there is
Memory.
the whole population shifts, but individual cells don't change their ranking, is essentially what it looks like. Oh, this is probably the wrong way to do it. Sorry if anyone has prone to seizures. So yeah, this is the activating the retina.
Yeah.
So we see non-uniform activation, but we see uniform depletion of ATP when we activate the retina.
Mhm.
And conversely, if we silence the retina, we see relatively uniform calcium levels as a population, but the ATP levels don't really shift from that, right? There's a little bit of an uptick, very slight, but the rankings seem to stay pretty much the same.
And so with optic nerve crush, you're seeing greater ATP levels, and what is the effect on calcium levels?
Calcium levels in the cytoplasm don't change. Calcium levels in the mitochondria slightly lower is what we're observing there.
Yeah. I think there are a lot of fun mysteries to unpack with this work, and I don't think I'll be able to, in a single viewing, do anything about them. But I really appreciate you showing them.
Yeah, no, we appreciate the suggestions.
And since you have all these really awesome sensors, are there any sensors for creatine phosphocreatine ratio? Or even phosphocreatine on its own? Because it's supposed to be kind of a reservoir for energy that's at equilibrium with ATP. But even when ATP is not changing, I had this sense that it was maybe a more sensitive sensitive readout of energetics. But I don't know for sure.
Yeah. So off the top of my head, I have not seen a sensor for phosphocreatine or creatine. I would say I haven't gone sensor shopping for a good couple years, so it's possible that there's some new ones available. But yeah, a few years ago, I basically just got as many review papers as I could about biosensors and got everything I could from Addgene or synthesized them. So I have like NAD and I have lactate and pyruvate and glucose and all this stuff, but I haven't seen a creatine sensor, but I wouldn't be shocked if one's available now.
Yeah. I think for some, actually, it would be really fun, which probably is not required for you to learn that much new. So don't do it. But you know, if you had this system where you use KCN and you shut down, and you're seeing this bounce back of ATP production, it would be neat if you would see a corresponding increase after KCN treatment of lactate production that didn't bounce back, that was sort of sustained. That would go towards this idea of switching after your inhibitor treatment.
Yeah, that's a very cool point. Yeah.
And then your subpopulations of ganglion cells that maybe, you know, you suspect are sustained glycolytic versus switch to glycolytic or whatever, you know, that would also potentially be supported with the lactate sensor.
Yeah. So the only downside is the lactate sensor is not the best. It has a pretty small dynamic range.
Downside number 2 is that lactate could be produced elsewhere and kind of come in.
Yeah. Yeah, so of the metabolite sensors, the glucose is the most robust we have. And as I mentioned, that just doesn't change. Like we've done a million different manipulations, and crush and death are the only ones we've seen change glucose levels in a cell.
Yeah. Well, when it was increasing after crush, I think, I was like, okay, yeah, energy demand is decreasing because it's not consuming its, you know, fuel.
Yeah.
And
Yeah, I think that could be part of it. Yeah.
Yeah. But I guess this data is not extraordinarily supportive of that.
Well, also, I mean, this is intracellular glucose as well, so that means that more is coming in. I suppose if you have a passive transporter and more concentration outside the cell, that you're going to get more in. The thing that we still don't understand is why it's different across the ganglion cells at rest and
Transporter
reproducibly
levels,
different.
are they different?
Sorry?
Are transporter, I mean,
Yeah, transporters.
steady state
Yeah.
glucose concentration is going to be, you know, dependent upon the rate of transport in and the rate of consumption. So if you find some difference in hexokinase level or isoform or transporter level or isoform, then those are, I think, the only explanations that could lead to this.
Yeah, we've gotten into all of the sequencing, and like the transcriptomics don't explain anything.
Yeah.
We're also wondering if there could be different types of transporters that are unexpected to be there that have different, you know, dissociation constants. The other thought is biogenic pathway shunting of glucose could be very different.
Yeah. One more thought to add to that would be that hexokinase is allosterically regulated by ATP, I believe. So it might just be that some of the data you already have is affecting the activity of the hexokinase.
Yeah, that's a very good point too that we've overlooked.
Okay. All right. We're over time, so I'm going to let everyone go. Thank you so much, Philip. This was a great talk.
Yeah, thanks again for having me. This is really fun. So, see you.